Alright friends, welcome back to Just Biotech. In today's lecture, we're going to discuss about the possible effects of chloroquine or hydroxychloroquine to inhibit SARS CoV-2 virus, that is the coronavirus that is killing the world right now. So let us first see the chemical structure of chloroquine and hydroxychloroquine. It is the anti-malarial drug that is being used for decades to treat the malaria across the world, and the basic difference between the structure of chloroquine and hydroxychloroquine is only the presence of this hydroxyl group. Considering the less drug-to-drug interaction and less toxic nature of hydroxychloroquine, this is the most preferably used choice to treat autoimmune diseases like rheumatoid arthritis. So this is the potential drug that is being tested clinically for the treatment of coronavirus 2 disease, that is COVID-19. So here are some possible effects of hydroxychloroquine on SARS-CoV-2 virus or the viral infectious cycle. The very first effect is as it interferes with the ACE2 glycosylation. As I told you, the first infection cycle step in the coronavirus 2 cycle is the entry or the binding of the viral particle to the ACE2 receptor, that is the angiotensin converting enzyme 2 receptor. What does the hydroxychloroquine or the chloroquine do is it interferes with the glycosylation of the receptor, and hence it prevents the binding of this viral particle to the target cells. The next effect of chloroquine is that it limits the synthesis of sialic acid. So there are multiple glycoproteins and gangliosides that are present on the surface of the cells that contain the sialic acid. Now, the coronavirus 2 is supposed or proposed to interact with these sialic acid with the help of its spike protein along with ACE2 receptor. Then, if chloroquine interferes or possibly limits the biosynthesis of this sialic acid that may be required for the cell surface of binding of coronavirus 2, it ultimately limits the viral entry inside the cell. Even after this, if some viral particles have found their way into the inside the target cells, what the chloroquine effect is, if binding of some viral particles is achieved. So what chloroquine do is it modulates the acidification of the endosomes, and hence it also inhibits the proteins cathepsins. As I told you in my previous video, that cathepsins are the proteins which are required for the release of viral particles as it further cleaves the spike protein inside. So it inhibits the chloroquine or hydroxychloroquine, inhibits the protein cathepsins, and it also modulates the acidification of endosomes and inhibits the formation of autophagosomes, which are required for the viral entry or the release of the viral genome inside the target cell. So these are the first three possible ways in which the chloroquine and hydroxychloroquine can affect the viral cycle. The very first is the glycosylation of ACE2 receptor. The second is it limits the synthesis of sialic acid. Third is it modulates the acidification of endosomes, or it increases the pH inside the endosomes, and hence it limits the formation of autophagosomes, and ultimately it limits the release of viral genome inside the target cells. The fourth possible way of the effect of hydroxychloroquine or chloroquine is it inhibits the phosphorylation of the mitogen activated protein. So what is the target is it inhibits the replication of the viral genome. And how does it inhibit the replication of the viral genome is by reducing the cellular mitogen activated or MAP kinase protein. How does it inhibit? It inhibits its phosphorylation, and MAP kinase are mostly required for the activation of virus replication. So when it is reducing the cellular mitogen activated protein kinase, it ultimately inhibits the viral replication. Besides this, there is again the last checkpoint which is offered by the drug, that is hydroxychloroquine or chloroquine. That is, it could alter the M protein, or it interferes with the proteolytic processing of M protein, and hence it interferes with the maturation of viral assembly and budding. So by interfering or by altering the proteolytic processing of M protein, it ultimately interferes with the assembly and budding of the mature viral particles that occurs inside the endoplasmic reticulum and the trans-Golgi network. It interferes in the post-translational modification of the viral proteins, that is the M proteins, and it interferes with the viral assembly and budding of the mature viral particles. So these are the 5 possible ways in which hydroxychloroquine or chloroquine can affect the SARS-CoV-2 viral cycle. The first one is it interferes with the acylation, glycosylation of ACE2 receptor, that is the angiotensin converting enzyme receptor. Second, or the B point, it limits the synthesis of sialic acid. Third, or the C point, is it modulates the acidification of endosomes, and hence it controls the release of viral genome inside the cell, target cells. Fourth point is it inhibits the viral replication by interfering with the phosphorylation of the MAP kinase, which are required for the activation of viral replication. And ultimately, the last is it interferes with the viral assembly and budding by interfering or reducing the post-translational modification of the viral protein or the M proteins. So these are the 5 major possible ways that could be utilized, or that could be the mechanism of action of the hydroxychloroquine on SARS-CoV-2 viral cycle. So thanks a lot for watching this video. Please share